Today in this video we are going to look at what is virus, what is viroids, and what is prions. These three different things. So we are going to differentiate each one of these into specific in details. So first of all, in general, as we know now, due to this pandemic COVID-19, we are all very curious about viruses and getting know to in getting know to viruses. So basically, in this video, we are going to give a general view about viruses, basic view, and also differentiate these three things. So what is virus? Virus is the smallest known to be say particle. So which causes diseases and only can be cured by vaccines or antiviral drugs. So vaccines are basically weakened pathogens. So vaccines contains weakened pathogens. Weakened pathogens meaning the weakened viruses. If so, in this for this COVID, there are various vaccines, but one of them uses weakened pathogens, meaning they will have the weakened virus, which is called SARS-CoV-2. Weakened SARS-CoV-2. How they can weaken the virus is that they carry out several steps, and one of it is that they will take out the spikes. So as the virus looks like this, okay, this will be the RNA strand inside, and this will be the envelope. The envelopes have spikes. So this one is basically coronavirus, crown-like spikes. Crown-like spikes. So what is meant by weakened pathogens or weakened viruses are included in vaccines is that they will take out these spikes. Why is that? Because the spikes is the one that makes the virus virulent and able to bind with its host. The SARS-CoV-2 binds with the cell specifically known as the receptor known as ACE2. So this ACE2 known as angiotensin converting enzyme. So that is where it got its name ACE2. So this is so that is why they are taking out this spike, so that our immune system knows, our antibodies or immune system known as white blood cells, they be triggered by the weakened pathogen. So it will cause a mild fever and mild effect of the virus, but then it's not too dangerous as though the real not weakened virus enters our body. So we are basically creating memory cells. So as we know, the graph of the immune, so this one will be immune and this one will be the vaccine injection. So this is why the graphs looks like this. So the vaccines will be injected 3 times or 2 times. So the first immune will be, it will rise, and then for a certain level it will drop, and then at this point this one will be the second injection. This one is the first injection, and then they inject and then it rises again, and then at some point this one will be the immunity level achieved. So this one immunity level achieved. So at this point it will go as person is immune to COVID-19. So what do you guys think? Viruses are living cells or non-living cells? Are they cells? So are they cells, and are they living or non-living cells? So this debate is still going on among scientists, that whether viruses are living cells or non-living cells. So what do you guys think? And if you guys think viruses are non-living cells or living cells, state it, put it in the comment, and state why you are telling so, like it is whether living or non-living cells. So virus, some scientists says is non-living, some says living. In what aspect they are telling it is living or non-living is that based on life. So what is life? If a living thing or life is told to be being cellular, first point, being cellular, then virus is considered non-living cells, because virus is not cellular and viruses are acellular. Viruses are acellular. And the second point, if life is defined as being capable of making own life through own metabolic system, then viruses are not living things. Reproducing using own metabolic system. So viruses are non-living things. And then if this one is the third aspect. So if life is expected to be defined as being able to specify each generation according to its own genetic information, then viruses are living things. So the third point, if life is defined as able to make generations based on own information. If life is defined to be this, then viruses are living organisms. So this is why the whole thing is being hotly debated among scientists till now. But then what they are now concluded, so it's not a final conclusion, but then for now it is concluded that the viruses are considered living if they are inside the cell. If the viruses are outside the cell, it is considered non-living or inert. Okay, let us move on to the discoverer of virus. So who first discovered virus? The person who discovered a virus named Beijering. So this was the first scientist who discovered virus in the year of 1897. The name virus, he is the one that coined the name as virus. So virus, what does virus means is that in Latin, virus means poison. So this was to this word virus was get to know or introduced by this scientist named Beijering in 1897, and this is the origin of discovery of the virus. So if you guys want to know who discovered the first vaccine, is that the scientist named Edward Jenner. This scientist discovered vaccine without knowing that is the vaccine. So this scientist is the one that created first known vaccine for which disease is that? Smallpox. Smallpox was very it was presenting very like COVID-19, which all over the world. So he was the one that cured smallpox by creating a vaccine. How he created the vaccine is that smallpox occurred among the humans, but then cowpox occurred among cows. So what this man did is that, okay, as we told just now, vaccine was weakened pathogen, right? Weakened virus or weakened pathogen. So this guy, after the cow had cow pox, pox meaning the skins have the pox, right? So the pox, after the cow have cured by its own cured. So the blisters, the cowpox blisters, these men scrapped the blisters, and then so meaning that the scrapped blisters of cowpox is meaning weakened pathogens. So now the viruses are no longer virulent or no longer very disease causing, critical disease causing, that state. So that is basically the scrapped virus particles of cowpox. So this guy made so these scientists made this scrap cowpox into a vaccine and made and cured cowpox. So this is basically the to be known basic history of discovery of virus and then discovery of vaccine. So next we are moving on to viroids. So what are viroids? Okay. So first of all, viruses are new infectious agent that is smaller than virus. So basically, viruses are new infectious agents which are smaller than viruses, normal virus. So it was discovered in 1971. First, they discovered the disease first, and then when they saw which is causing it, and then they found out viroid, which is very small than virus. So what was the disease? Is that potato spindle tuber disease. So potato spindle tuber disease. So potato spindle tuber disease occurred in potatoes. Like a normal potato looks like this, but then these viroids cause the potatoes to be look like this and then look like this. Looped. The potatoes were looped, and then the farmers were worried why our potatoes are being like this. And then what they did is that they mentioned, and then researchers were finding out what was the cause. And then they can't normally isolate. They were using techniques to isolate virus which are larger than viroids. They didn't know at that time, but then they went specific, and then they found that viroids only occurred in they have only RNAs, so there's no DNA in viroids. And also what they discovered is that now usually they are known as plant pathogens, and they won't produce protein. So that when the RNA is when the RNA is being what we call transcribed by RNA polymerase. So viroids are specifically transcribed by RNA polymerase 2. RNA polymerase 2. So these do not produce proteins. So from RNA, usually viruses and also other cells makes proteins, but here does not produce proteins. And another thing is that viruses are very small and the RNA is very small, and the length of the RNA is only 300 to 400 nucleotides. And the RNAs are always short because it's just 300 to 400 nucleotides, circular, and highly complementary. Another point is that these viroids do not have protein code. Normal viruses do have capsid, which is known as made up of capsomeres. That is known as protein code. But then viroids do not have protein code. Only made up of RNA. So they lack protein code. And also no protein code, and no capsid. Only exists as naked RNA. Another point is that these viroids do not code, do not have AUG codon. AUG codon. So these are basically about viroids. And you guys want to know about the discovery of viroids is that okay, let me do here. So who discovered viroids is that it was Steiner. The person who discovered viroid is Steiner in 1971. In 1971, and the first viroid was named as PSTV. So you guys will be wondering what is PSTV. So as we know, the first viroid they discovered is potato, spindle, tuber, and V stands for viroids. So that was the first viroid discovered by Steiner in 1971. So lastly, we are going to move on to prions. So what are prions? Prions are coined as proteinaceous infection particle. Proteinaceous infection particle stands for prions. So okay, what do prions do is that prions are not considered as virus, but then there are some scientists telling that prions are virus, but then they haven't discovered it as in detail. So there are no proper evidence telling that prions are virus. But then some scientists tell that prions are proteins, some kind of proteins that misfolds normal proteins. So if they are misfolding normal proteins, then the functions are going to be lost. So the normal proteins are going to lose their normal functions. So what happens is that if the protein is enzymes, or normally proteins are enzymes, so if the enzyme they are losing the shape or losing the active site, then automatically they are going to lose its function, and then this causes metabolic problems in our body. So what basically prions causes what types of diseases is that, so it normally causes diseases in neural system. So first of all, it causes BSE, diseases that it causes is that BSE. So BSE stands for bovine spongiform encephalopathy. So bovine spongiform encephalopathy, also known as mad cow disease. So this occurred in cows, also known as mad cow disease, whereby the cows will be trembling, having tremors, and then the spongiform is known for it's the brain. Sorry, it's not brain. So the brain, it is normally compact with neurons, but then when they see for the patient having BSE, or the cows having mad cow disease, which is same, so they will have spongiform. Spongiform meaning they will have developed holes in their brains. This causes them to go crazy, and also cause them to have tremor, and then finally death. So the next disease is that CJD. So this is disease known as Creutzfeldt-Jakob disease, which occurs in humans. And then the last one will be fatal familial insomnia. So these are the diseases caused by prions. Okay, now let us look at the symptoms of prions. So, symptoms that we are having prions in our brains and in our body, basically. So, the first symptom is that they will have a difficulty in speaking, difficulty in speaking, and then dementia. Dementia is something that is difficulty in learning. Uh, it's hard for them to learn. The learning process will slow down. And then another symptom is that they will have confusion. This kind of thing will start confusion, fatigue. This comes under this confusion, fatigue, and then muscle stiffness, muscle stiffness, and then so if stiffness, this causes also difficulty in walking, difficulty in walking. So this is where the patient, the person who's affected by prions, they will first stop walking, and then finally the mobility will decrease, and then eventually die. So this is final stage. And then they will also have hallucination. Since hallucinations. Since they are having problems in their neurons in brains, they will start having hallucinations. So these are the symptoms basically when prions are affecting our neural system. It is very hard to cure this disease. The only reason is because prions are not like viruses and viroids. They have very specific characteristic, which is they have high resistance to heat, unlike viruses and viroids. High resistant to heat, high resistant to heat, ionizing radiation. So we can't use the normal things that we use for cancers. The radiation using radiations to cure cancers. We can't use that. So they have high resistance to this heat and then radiation, and also UV radiations. Also chemicals like phenols, which are normally used to treat diseases. So they have plenty of special characteristics, benefits that makes them to be having a comfort zone in our brain, in our body. So that is why it is hard to cure diseases caused by prions. So how they identify that these diseases are caused by prions is that by having MRI scans, samples taken from fluid of spinal cord, blood test to see the presence of prions, and then finally electroencephalogram, which basically electroencephalogram, the technique used to study brain waves. So they compare with the normal brain waves. If there is abnormal waves, then there is abnormal waves that are similar to the patients having disease caused by prions, and then they will determine that patient's having one of the diseases caused by prions. So now, as we have looked into viruses, viroids, and prions, so we are now going to differentiate three of them. Okay, so these are the points that we are going to look at in order to differentiate these three virus, viroids, and prions. So first of all, virus. First of all, virus. The genome that viruses have are DNA or RNA. Some viruses have DNA and some viruses do have RNA. And what about viroids? We saw that viroids do not have DNA, they have only RNA, which is very short RNA, 300 to 400 nucleotides. And for virus, it is 250,000 nucleotides. 250,000 nucleotides. It's either DNA or RNA. And prions, for information, prions do not have genome. They do not have genome. And strands. So strands meaning that they are single-stranded DNA or RNA or double-stranded DNA or RNA. So first of all, viruses are maybe double-stranded or single-stranded. For viroids, they should be single-stranded. For prions, it should be single-stranded. Single-stranded. And for code, so it's whether protein code. So viruses do have protein code. Protein code. But then viroids do not have protein code. As you guys remember, it's just naked RNA. So they do not have protein code. But then only one type of viroid do have protein code, which I didn't mention just now, which is HDV. Hepatitis D viroids. And then prions, they do not have protein code too. So the target. So what types of cells and what types of systems that these viroids and prions will affect is that target host, everything like eukaryote. Under eukaryote, as we know, it will be animals, plants, and so on, and also bacteria under bacteria kingdom, and also archaea. And then viroids. So viroids, what do they target is that just now we looked into plant. So viroids is basically, if we tell viroids, you should think of plant affecting as cellular organisms. So they are basically, they affect plant cells, only plant cells. And how about prions is that we do not basically have host for that, but then we can tell system or organs for prions. It's either brain, neural system. So this is target for prions. So this is basically the differences between virus, viroids, and also prions.